Welcome students. We have dealt with this how to make monohybrid cross as well as dihybrid cross. Today in this class we will see how to make the trihybrid cross, that is crossing two plants with three characters. Here crossing two plants to check for three characters. In monohybrid cross we were checking for one character, in dihybrid cross we were checking for two characters, in trihybrid cross we are checking for three characters. So we are checking for three characters. Now in this is a plant with these three characteristics. One is TNT, that is tall. First characteristic is tall, second is purple flowers, and third is the flowers are axillary in position. Position of the flower. Which characters we are taking? Here one is height of the plant, another is color of the flower, and third is position of flower. Third character which we are taking is position of the flower. So here axillary position is the dominant one. In this plant we have three characters, that is the plant is tall and the color of the flower is purple and the position of the flower is axillary. Okay, and we are taking another plant with other three characters, that is this plant is having dwarf, this is dwarf, the color of the flower is white, and the position of the flower is terminal. The position of the flower is terminal, that is at the end of the modified, that is stem, floral bud. At the end of the floral stem we will get the flower. So three characters, these three are total contrast with these three characters. So a plant with contrasting characters, with three contrasting characters we have taken. Okay, now this is the parent generation. This is the parent generation. We are crossing two parents to check for three characters. We are taking two parents plant to check for three characters. So the parents will first produce gametes. The parents will first produce gametes. So when it produces gametes, now we get this T will be with this P, this T will be with an A. So when we cross this, when we see this, now we get gametes of this genotype. We get gametes of this genotype, that is capital P, capital T, capital P and capital A. We get 8 combinations. We get 8 combinations of this one. As it is difficult, I have already explained you how to make this, how the gametes are formed in the monohybrid cross and dihybrid cross. So you can do it in your book, you will be able to do it. So here in this now we get 8 gametes of this capital T, capital P and capital A from this plant, and from this plant again we also get 8. Here all of them are dominant and here all of them are recessive characters. All of them show recessive characters, they have recessive genes, alleles, and this have dominant alleles. All of them are dominant. So we get 8 gametes of this combination. 8 gametes of small t, small p, small a, and we get 8 gametes of capital T, capital P and capital A from one parent and from another parent. So we get 8 gametes like this. So if we make a cross, so this 8 at one side and this 8 at another side. If we make a cross with this 8, TP, for example, write like this. I'll write here TPA, 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 TPA. So like this 8 you will get, 8 at one side and this 8 at other side, this 8 at other side. So this is, so if we plot this 8 on this 8 on this side and this 8 on this side. So in the first generation we will get the probable combinations of this. In the F1 generation we will get the probable combination with this one capital T, small t, capital P, small p, See here, capital T, small t, then capital P, small p, capital A, small a. So like this, because all of them are same here also, all of them are same here also, we get the same gametes in F1 generation. This we have seen in both monohybrid as well as dihybrid. F1 generation, all the gamete, all the offsprings are same. So here we get 64 offsprings. If we make the cross now, in trihybrid cross we will get 64 probable combinations. In monohybrid cross we were getting 4 probable combinations. In dihybrid cross we were getting 16 probable combinations. Now in trihybrid cross we will get 64 probable combinations. 64 probable combinations we will get. So what type of 64? That is 64 combinations, of which capital T, small t, capital P, small p, capital A and small a. This type of offsprings. How many offsprings of this type? 64 offsprings of this type we will get. We will get 64 offsprings of this type. You can do it in your book. On this board it is very difficult to do it. So you can do it in your book. Already explained you how to make the gametes. But we will get this gametes 8 and this gamete 8. So if you cross it now, if you cross it, because all of them are same, you will get the same combination. So in the F1 generation, the phenotypic ratio, in F1 generation, phenotypic ratio will be 64:0. And genotypic ratio is also 64:0, because all of them will show tall, purple and axial flowers. Because dominant in F1 generation, whatever we get is dominant traits. So all of them, if we see this one, which height will be expressed? Tall height. Which color will be expressed? Purple, because purple is dominant. Which position is expressed? Axial, because axillary position is dominant. So all of them are tall, purple and axillary flowers. All of them are tall, purple and axillary flowers. So this is heterozygous, not homozygous, because it is having one capital and one small, one capital and one small. So all of them will be tall, purple, axillary. Hetero, all of them will be heterozygous, tall, purple and axillary. So now this was about the first generation. If you want the second generation, you self this one, any selfing with any two, this with any, that is out of 64 you can combine it, that is cross with anyone. So first and this one, these two on selfing you get F2 generation. When you self this, we will get 64 now, out of 64 you take any two and cross it. So this is called as selfing. So when you self it, we get the F2 generation. We get the F2 generation. In F2 generation, first these are the parents. These two are the parents. Now gametes will be formed. What type of gametes will be formed? When these two are crossed now, this will form gametes like this. First is capital T, capital P, capital A. This also capital T, capital P, capital A. This is the first type of gamete. Then next type is capital T with capital P and capital T with small a. Here this one and with small a. So this is, in this all three are dominant. All three are dominant. Here in this now two are dominant. Here also this is, these gametes will form. This will form these gametes, because both of them are same gametes will be formed. So in this, in the first type of gamete, all three are dominant. In the second type of gamete, that is capital T, capital P and small a, two are dominant, one is recessive. And the third one, here also capital T is dominant, capital A is dominant, capital P is, small p is recessive. So here also two dominant, here also two dominant, Here you see, here also small t is there, one dwarf, and here these two. Here also two dominant. Then we see here we get one dominant and two recessive. Only T is dominant, this P and this A is recessive. 2 is recessive. Here also one dominant, two recessive. Here two recessive, one dominant. Here all of them are recessive. All of them are recessive. So these are the gametes. When you self this na, it will produce gametes. What type of gametes? These 8 type of gametes will be formed. It will produce these 8 types of gametes. So this will also produce these 8 types of gametes. When we self these two na of F1 generation, we will get gametes of, for this we get this gametes, and for this we get this gametes. You can see, you can calculate, you can do it in your book. If you do it now, you will be able to get it. So we get this, that is gametes will get when we cross this, when we self this in F1 generation. These gametes we get in the F2 generation. So now coming to this, how to calculate the genotypic ratio and the phenotypic ratio. How to calculate the genotypic ratio and the phenotypic ratio, because it is very difficult to calculate over here. So we have the formula to calculate the phenotypic ratio. The formula is 3:1 to the power of n. So and to calculate the genotypic ratio, the formula is 1:2:1 to the power of n. So in F2 generation we get what we get. One is, you just first generation. If we take for first generation, here 3:1 into first generation means 1. F1 is 1. So 3 ones are 3. Here 1 into 1, 1. 3:1 we get. So in this genotypic ratio also 1:2, 2:1 to the power, which generation it is? This is the generation. This is the generation. Which generation? First generation. 1 into 1 is 1. 1 into 2 is 2. 1 into 1 is 1. So in F1 generation, the phenotypic ratio will be not first generation. Wait. This is the number of characters. This is the number of characters. Here also number of characters. So in monohybrid, this is for monohybrid. This is for monohybrid. This is for dihybrid. Arey wait. This is for phenotype of monohybrid. This is for genotype of monohybrid, because how many characters we are taking? 1 characters. This is the phenotypic. This is the formula to calculate the phenotypic ratio of monohybrid cross. This is the formula for calculating phenotypic ratio, and this is the formula for calculating genotypic ratio. So for F1 generation means we will take not first, for that is monohybrid cross we will take 1. For dihybrid cross you take 3:2 and 2 for dihybrid cross. Monohybrid ratio for dihybrid cross. Then this is the general formula for finding out phenotypic ratio. This is the general formula for finding out the phenotypic ratio. So if we want to find out the phenotypic ratio of monohybrid cross, then instead of 1, mono means 1 character na, we will be putting 1. Okay. If we want to calculate the phenotypic ratio of that is dihybrid cross, 2 characters, then instead of 1 we take 2. if you want to calculate phenotypic ratio of trihybrid cross, we will take 3:1 into 3 because it is for trihybrid. General formula for phenotypic ratio is, to find out phenotypic ratio is 3:1, and here n to the power of n denotes the, that is monohybrid, dihybrid or trihybrid cross. We will study this, we will continue this again in the next class, where I will show you how to calculate this phenotypic ratio as well as the genotypic ratio.